Hi, everybody. Thank you for joining us today, and I appreciate the opportunity from the organizers for allowing us to speak on this topic. I'm Max Sellman. I'm our gene editing product manager here at Aldevron within our protein team. Thought we'd mix it up a little bit, have a couple different speakers. As you'll see, Jim and I kind of focus on different areas, and they tie together really nicely. And so kind of excited to just give you a little bit of overview about what Aldevron's been doing in the gene editing space, and see how perhaps we can help you out or work together as you move forward in your programs. So just kind of as a way of background, level setting, a little bit about Aldevron. We're a biomanufacturing company. We've been in the industry about 25 years. And kind of simply put, our mission is to be the supplier of choice in the cell and gene therapy space. And so, these days we can't even keep our slides updated with how many employees we have. We're up to nearly 1,000 employees across a few different sites, primarily in Fargo, North Dakota, and Madison, Wisconsin. In terms of what we make at Aldevron, what we like to say, we manufacture the central dogma of molecular biology, right? So whether it's plasmid DNA, research grade through GMP, mRNA, research grade through GMP, or protein, research grade through GMP, these are the areas we serve. And again, primarily the cell and gene therapy space, but really a wide variety of applications in all of those areas, as you can imagine. What we wanted to talk about today, and kind of the thrust of what we'll be talking about, are kind of gene editing proteins and how Aldevron has approached gene editing, and kind of the products we offer, and some of the solutions that clients have taken advantage of to accelerate their programs. So. By way of background, when we think about CRISPR nucleases kind of in our catalog, and I should say a lot of what I have is focused on nucleases that Aldevron manufactures and sells as catalog products, but we do have a really long history of manufacturing custom nucleases, custom nucleases, base editors, nicases, things like that. But I'll kind of focus in on the catalog and how some folks have been using that, show you some data of how these have been applied. When we think about nucleases at Aldevron, everything we think about is how are we going to get folks to the clinic and have a nice seamless transition to there. So what you'll see with all the different proteins that we have on offer is a really consistent, clean construct design. So across all of these products, all you're going to see is the nuclease itself, which of course is going to vary based on the application, and then an N-terminal and a C-terminal nuclear localization signal. And we keep the construct very simple. The idea is less is more. You don't have to explain other features of a construct to a regulator when you're thinking about a clinical program. You have kind of the bare necessities to get your protein into the cell and achieve gene editing. And so, I'll really zero in on these top proteins. I'd say these top two have been kind of the most popular historically, and there's kind of the biggest body of data around them. We are really excited to launch a new protein as well. And so I mentioned earlier when we think about research grade through GMP. So Aldevron was the first company, it's been a number of years now, but to launch CRISPR nucleases as an off-the-shelf catalog cGMP product. And so the intention here was to have very high quality products that are available for immediate delivery that are suitable for a clinical program. And not only to have them in stock, but to actually have them at GMP in an identical format to the research grade product. So when you transition from research into the clinic, you can transition to a GMP reagent. You're not going to get better performance. You're not going to get worse performance. You're going to get that same level of performance because it's the same nuclease. And so we've been doing this for several years, and we've, as things have progressed, we've continued to build on this offering. Where we are today, these products have been used in easily more than a dozen clinical trials to date. We also have drug master files for these, so that's always been kind of a challenge, is how do you incorporate the manufacturing information from your third-party manufacturer for protein, which is going to be looked at kind of like a drug substance in the context of an ex vivo cell therapy. That's sort of the level of rigor that the FDA has been asking for those therapeutics. We have that all wrapped into master files, so it's super easy for clients to just reference that file as part of their IND process. And so I'll talk a little bit about the newest product, and then I'll show you some data, which is kind of the exciting part. So this is the newest thing. We're very excited about this. We're going to be launching a product here in the next few weeks, which is based on Inscripta's MAD7 nuclease. So we're really excited to have this in the catalog. We will be taking this product to GMP next year. It's a catalog product off the shelf. What's really great about this is it's bringing new features compared to the other GMP nucleases that we already have. So this is a type 5, class 2 type 5 CRISPR nuclease. So it's going to behave as those canonically do, right? It's going to have a T-rich PAM that it targets rather than the G-rich PAM that an SP-Cas9 looks for. It's going to create that staggered cut rather than a blunt double-stranded break. And what's interesting about this product is that it exists as separate IP from what's claimed by some of the other folks out there that, for example, have like a Cas12a in their catalog. So the MAD7 nuclease is very distinct from a sequence perspective from some of the other tech that's out there. And as we mentioned, we licensed this from Inscripta, who's the group that discovered it. They're based out in Colorado. They're a really good team to work with from a licensing perspective. And so, we're really excited to have a product that just by purchasing the product, you don't have to have any sort of licensing deal with Inscripta. You can just buy the product off the shelf and use it all the way through clinical trials. And so, the plan here, like I said, is to have this available just in the coming weeks. I actually have some available on kind of an early access basis. So if there's folks that are interested in MAD7 or knowledgeable about it, interested in applying it in their research, that's something that we're really excited to be able to support. I have these in a little bit of a funny order, so I'm going to actually jump back and then jump forward. So that's the MAD7 product. What I'm going to spend a lot of the rest of the time on, and when I throw it over to Jim, a lot of his focus on is on this SPI-Fi Cas9 nuclease. So I'll talk a little bit about this. So this is a product we've had in the catalog for about 5 years now. You might be familiar with our sister company within Danaher, IDT. They, in 2017, published this paper, which I'd be glad to direct you to, with Matt Porteus's lab at Stanford. And to summarize, what they found in this paper, that a single point mutation in SP-Cas9 sequence, an arginine to an alanine at position 691, has the effect of nearly eliminating off-target effects in just about every case that they've looked at, going from some measurable level of off-target effects and driving that down to background type levels. And so, kind of the next few slides that I have, I should say this is a product that, we're carrying an off-the-shelf GMP product. It's been very of much interest to be used in clinical trials, as you can imagine, with a reduced off-target profile. And so I'll just kind of show you some data that we've plucked from various sources, various partners, collaborators, that really kind of show off the power of this nuclease. So, briefly, what we have here, just some really simple data, just looking at knockout in iPS cells. So we're working here with a company called Vascugen, which is also based with us in Madison, Wisconsin. And Vascugen works on iPS for cardiovascular disorders. And one thing that they were interested in doing is just having a really kind of consistent workflow where they can, batch to batch, lot to lot, cell lot to cell lot, have kind of consistent performance. And they were really happy to report that even before doing a lot of optimization, they're getting kind of an average knockout in the range of 88% in their iPS cells, really nice kind of consistent results at their target, which they're not disclosing here, well below the line that they've kind of determined, where they came into their experiments and said, look, if I can get below 30%, basically, of cells that are expressing the protein of interest, I'm going to be happy. We can carry this forward with our process to end up with a pure final product. So, an IPS, kind of robust editing solution. Drilling down a little bit more, next couple slides, we did in collaboration with our partners at Maxite. Shout out to Jim. And so what we did here, you might be familiar, there was a paper a few years ago that was published by Edward Stabmauer and Carl June, where they looked at doing a multiplexed knockout in T cells They were looking at kind of the safety of creating a gene-modified CAR T, where they would do a triple knockout. They would knock out the T-cell receptor as well as the PD-1 receptor. It's a really interesting paper. If you're not familiar with it, definitely take a look. The big question that they were trying to answer essentially is safety and efficacy around gene-modified CAR T. And what was interesting from Aldevron is we were reading this paper and we saw that there's some reference made to off-targets. It was something that he looked at was frequency of off-target editing, and made some comments to the notion that there are new tools, and they actually cited to that paper I showed you a minute ago, that there are new tools out there that could reduce your off-target editing. And so we thought it would be an interesting project to basically recreate the work that was in that paper, and then take a look at kind of on-target and off-target editing performances using some of these nucleases that I just showed you. And so what we did here was go in, we took the guide sequences from that paper, had them synthesized, went into T cells and did our gene editing, again using the Max electroporation technology to deliver the gene editing nuclease. And then the question was essentially, did this work, right? That's the first question. Were we able to get robust knockout of the target receptors? And by flow, the answer to that is yes, absolutely. You're able to go down to certainly less than 1% for one receptor, kind of 1% roughly expression on the cell surface of those receptors. And I'll note, these experiments weren't super optimized. I mean, we spent a few times, we looked through a few permutations, but this is a lot closer to first pass than, let's say, an optimized T-cell editing process. And we were able to get really robust, practical knockdown of the expression of T-cell receptor as well as PD-1 receptor. What was really interesting to me, though, looking at this data, and I should say I'm not showing the data, but you're getting indel rates consistently, 95, 96, 97% in these experiments. And I'll note as well, very consistent results here when you look at using a wild-type SP-Cas9 versus using the SPI-Fi Cas9, right? Virtually identical in terms of on-target performance and practical knockout of those genes. What's really interesting is the off-target data. And so what we did here is, again, looking into the data from that paper, you could see that they had done some off-target analysis and had identified some key potential off-target sites. And there were a couple of them. These two really stood out. It was annotated as CLIC2 or LDAH. Those are both off-target sites that were actually in the coding region of genes. And so the thought was, let's take a look at those, see what the effect would be of literally just changing. The only thing that is different in these experiments is the nuclease that was used. In the orange bars here, you've got a wild-type SP-Cas9, whereas the kind of gold-colored bars here is the SPI-Fi nuclease. Everything else is controlled. And what we found, and I should say the methodology for doing this essentially is you do your gene edit. We do PCR generation of amplicons at these sites, and then you do next generation sequencing and take a look at your proportion of reads that are coming up as off-target edited. And in some cases, there's no difference. And I think we know this, right, from other CRISPR research that sometimes with good guide selection, a wild-type nuclease can be very effective, and you can have pretty limited off-target editing. But in other cases, you do get off-target editing, and it is a concern. I mean, in this case, at this site, you're seeing nearly 4% of reads coming back as having an off-target edit. At this site, you were seeing about 2% of reads. What I hear from clients really consistently is they want that number to be like 0.1%. Or lower, as low as possible, certainly, as close to background as possible. And just by substituting in SPI-Fi Cas9 for the wild-type Cas9, you get knocked down to background level for your off-target effects, because the blue bar here, we didn't electroporate those cells. So this, to me, is a pretty powerful indication of the power of that SPI-Fi nuclease and how that one mutation in the sequence, which is the only difference between those two constructs, that arginine to an alanine, has that result of nearly eliminating off targets. And this is just kind of a snapshot of data, but we've really seen this borne out just about every experiment, just about everything that's been published around the SPI-Fi nuclease, is that it's a really robust tool for eliminating off targets, cutting them off at the source. So, that's just a there, SpyFi, right? So that's just a little bit of the data. I'm going to pass it over to Jim. He's going to tell you a bit about nanoplasmid. So one really excellent, one question I should say around gene editing, of course, and using CRISPR is to the extent that you're going to do knock-in, how are you going to deliver your HDR template? And there's a lot of different technologies out there. Jim's going to tell you a bit about Nanoplasmid, which is kind of the newest offering within Aldevron's program, show you a little bit more data combining power of nanoplasmid with this nuclease, and you get pretty excellent results. So I'll pass it on to Jim, but thanks for listening.
So Nature Tech's been around almost as long as Aldevron has, and we focused on a very different thing than Aldevron. We're a vector design company, and we developed a nanoplasmid vector platform. We offer research-grade services, and so it was really logical for us to be integrated and be acquired by Aldevron, because now we offer the GMP services that go with that. So that was something that the clients always wanted, start to finish, and we're always having to try and marry them with a CMO. Now we have that start-to-finish experience. So I'm going to talk about the nanoplasmid platform. And I'm going to give you a background of what the nanoplasmid platform is. Some of you aren't working on CRISPR, but it has benefits to a wide variety of vector types. I'll go over that briefly. And at the end, I'll talk a bit about the use of nanoplasmids as donor HDR templates for knock-ins. Okay, so basically, most people work with plasmids that were actually developed in the backbones, 1980s, puck plasmid, most of those things that you see out there. When you're dealing with a AAV vector, a lenti, or a transposon, gene therapy vector, if you think about your backbone, it's always got like puck origin with an AMP gene or a CAN gene. And those are big, they're like 2.5 or kb or larger, and they provide the bacterial functions that you need to replicate and select an E. coli. And virtually no research was really done on that since the 80s. That's why you're still using them. Most people focus on their payload. If you're a virologist, you focus on between the, the ITRs or the LTRs. And so we focused on the backbone. And so we ended up with a nanoplasmid platform in 2 steps. The first step was clients or biotech companies were coming to us with the concerns about antibiotic marker in the backbone. There's a lot of regulatory guidance that obviously, especially in Europe, absolutely hate having the markers in the backbone. There's a lot of pushback against having that. And so we were approached to, can you get something that's not going to have an antibiotic marker or a protein, which also creates a lot of regulatory concerns. So that was the first piece of it, was to make the RNA out part. And all that is a very short RNA, and again, it has no coding capacity or any kind of antibiotic resistance. It simply is an RNA that regulates expression of a chromosome-encoded selectable marker, sucrose selection in this particular case. The whole thing is about 150 base pairs long. The next problem that we tried to solve was that with people who were working with liver gene expression, and they were getting silencing of their vector backbone. And what was found by work by Mark Kay's lab at Stanford was that large backbones lead to silencing of the vector when you deliver it to the liver. So we worked on replacing the large puck origin with this small R6K origin. That was the only way we could get the backbone small enough not to have gene silencing. By switching to the R6K backbone, we also had to develop manufacturing technologies, because the R6K is different than the puck origin, and is that the R6K origin requires a trans-supplied replication protein. So we had to develop cell lines to actually provide that in trans to allow you to replicate this plasmid. So we named the plasmid that resulted the nanoplasmid platform with this small backbone. And we got expected effect of long duration. That was what we're supposed to be doing. We got around the gene silencing. And of course, we had no antibiotic markers. What was unexpected was the expression was way higher than you would get with a mini-circle or another type of vector platform like the plasmid. And then we had very low toxicity. These 2 attributes, high expression, low toxicity, were not expected from this. the work that had been done at that point. So now we kind of have a good handle on, you have less cell transfection-associated toxicity. So you look at transcriptome analysis of cells that are transfected with plasmid or nanoplasmids, you see that the plasmid transfected cells have a very large transcriptome changes, whereas the nanoplasmid has a very light transcriptome change. And the improved expression probably due to less vector inactivation. A large backbone vector is identified as like foreign DNA by the sensing pathways, for example, that would sense a DNA virus coming in, and they're heterochromatinized and activated. And I think what was unknown at the time was just how much plasma was inactivated. So those two attributes led to benefits across the board for a variety of different platforms. So the gene therapy ones, that's pretty obvious. You're increasing the expression level and duration. That can give you, especially with the non-viral field where the therapeutic levels have to be pushed and you have to put more and more DNA to try and get to the therapeutic levels, that increased expression level allows you to increase your efficacy and potentially reduce your dosage that you need to get where the efficacy you need. For cell therapy, big effects here, both associated with this transfection toxicity and less vector inactivation. In both transposon vectors and CRISPR vectors, that step of putting the DNA into the cell, the nucleofection step of the primary cell, is associated with a lot of DNA-associated transfection toxicity that you get with a plasmid, and this is reduced a lot. So your cells are more viable, and they have better, they're better killing functions, as you would imagine, because they're not as hurt by the transfection effect. And other thing is your manufacturing timeline is reduced, because without taking a big hit in viability, you get back to the number of live cell count that you need. The FDA would mandate, for example, to infuse back into the patient. And then, because of the less vector inactivation, you're getting enhanced transposition in the case of a transposon vector and enhanced gene editing. And I'll give you an example case study at the end of the gene editing efficacy. The other one here, lentivirals and AAV, these are interesting ones. If you come to us with a lentiviral vector or you come to us with an AAV, we'd retrofit the vectors you use to make those things. Usually with lenti, there's 4 vectors. We would take your plasmid backbone out and put the nanoplasmid in, AAV, the 3 vectors. And when you put those into the HEK or whatever producer cell line you're going to use, the virus you make is exactly the same as the virus you gave us with the plasmid. So you can see the virus is not going to be changed by it. But the benefits are, again, this less inactivation. You get enhanced manufacturing titers with a lentiviral vector. With AAV, the single factor that I indicate here that is of interest to the AAV folks is this elimination of marker transfer to the patient, because the AAV has ITRs on the ends of it, and it can be reverse packaged, and about 1% to 2% of those viruses have reverse packaged the backbone. And so those have then kanamycin or whatever marker you had in the backbone. We eliminate this with the nanoplasmid, which has no coding capacity and no homology to the human genome. We can also get higher manufacturing yields, and that's just a function of the way that the system works, in that the puck origin has a temperature-sensitive replication origin, and it goes from a lower copy at 30, and you can increase the copy number. But there's a limit. You take it to a certain temperature, and that's the max that you can get in terms of the copy number. Because the R6K plasmid is driven by a rep protein expressed from the chromosome, we had the flexibility to ramp that chromosomal expression up as high as we want it. Allows us to get higher manufacturing yields than you would with a puck origin, because we don't have a cap on the copy number we can make. One last advantage of nanoplasmid is that thing I was just talking about, where you have to have a protein from the chromosome. So nanoplasmids are made in an engineered strain that allows for the antibody-free selection and for the R6K replication. So one of the issues with the puck origin is, I think the term would be promiscuous replication. If the puck origin gets into a gram-negative bacteria, it'll replicate. So you see a lot of reports in the literature of a manufacturing facility making a certain transgene, and then it gets into the environment outside that facility because all it has to do is transfect a cell and it replicates. Same is true of deliveries. If it gets in contact with a gram-negative, either through your gut or through the skin, it can replicate, and now you have your CAR or whatever it is you're trying to use as a therapy out in the environment. You don't have to worry about that with nanoplasmids. So we feel like, and I haven't really talked a whole lot about mini circles, but the work of Mark Kay's lab that I said before about the gene silencing, that was with mini circles, which is a way of making a vector that has about 100 base pair backbone. And so you basically grow it as a full plasmid, so it's got a marker and a puck origin, and then you do an in vivo recombination. Great research tool, very difficult to translate to the clinic. So this is considered to be a replacement for that type of vector by combining some of the things that you have with the mini circle with other advantages that you have with the plasmid, and then other things that are basically nanoplasmid specific. And when I talk about that surprising increased expression, it can be very dramatic. This is a group from Old Dominion. There's a number of groups that have been using our vectors and published on it. But you can see this is comparing a nanoplasmid expression in vivo to canonical plasmid, the traditional plasmid. So this is an exception when I mentioned most plasmids are from the 80s. This is a more modern one from the 90s. That's when the vectorology was done with, in the DNA vaccine field. But you can see there's just a dramatic improvement in expression. So I'm just going to end by sort of coming back to Cas9 and CRISPR templates, that the nanoplasmid platform works really well for HDR editing knock-in, and it works, I'll show you a case study here, works better than you would get with a standard plasmid or a linear DNA. And not just for knockout, you have lower toxicity, so you end up with a higher viability cell after the transfection and higher numbers of actual edited cells. And then, of course, you have that, as you know, the other points I talk about, the regulatory advantages, no markers, you're not going to change anything in terms of the integrated transgene, no replication because of the type of vector it is. So the case study I'm talking about here is from Genentech. They did a very extensive, looked at multiple different knock-in transgenes, multiple different target sites in the genome with both linear, plasmid, and nanoplasmid vectors. And they were using Cas9 ribonucleoprotein complex to integrate into the genome. And the key thing is on this side, this is basically the percent edited cells, and this is the nanoplasmid on the bottom. You got the plasmid DNA in the middle, and then the linear DNA on the end. And they were getting two times as much edited cells compared to puck, and 3 times as many as with the linear double-stranded DNA. So this was across a wide variety of things showing the nanoplasmid had improvement here. And this is kind of back to the, talking about how one of the things we liked with getting acquired by Aldevron was being able to offer vectors from start to finish, now having the GMP. We also can add now its component, this SpyFi Cas9, so you can get your HDR template from Aldevron. You can also get the Cas9 that you need. So basically, the nanoplasmid vector technology is built on the foundation of the vectors that were developed in the 80s and the 90s, and combining advantage of antibody-free selection and very high transgene expression that you get by having a very small backbone, and improves the performance of a wide variety of vector platforms. Okay, Max and I will take questions now.
Does anyone have any questions?
Thank you. I have a question about the nanoplasmid manufacturing. How tolerant are you of long regions of homology or repetitive sequences or very GC rich or GC variable regions?
So that's a great question. So we worked with AAV. AAV was the one that came along that had ITRs, which have inverted terminal repeats. And we had the same problems everyone else has with terminal repeats. But then we developed, which I didn't talk about here, the reviver host cell line. So we developed a high yield manufacturing cell line that allows us to deal with structured DNA. And so we use that for all our messenger RNA vectors, very stably and high yield produced like a poly-A vector, and we use it with all our AAVs for the ITR. GC, we really haven't had a problem with that per se, but yeah, we did have to develop a modified strain to be able to handle poly-A and ITRs.
Well, just to spice it up, I'll ask a stupid question. Have you tried using the nanoplasmid for increased expression of, say, biologics in CHO? Does it work?
So expressing biologics in CHO, it's not been a major application. Yes, it works, but a lot of people are using like integrated cell lines and stuff like that. It probably would be in those instances where you want to do a transient. I've seen that some people are doing transient expression in CHO. It would probably work really well for that. But yeah, there's so many integration systems that people use to get a stable cell line, so it wouldn't have application for that latter one.